In the healthy state, there is a mucosal barrier that consists of mucus, it consists of intestinal epithelial cells and IgA dimers. There are also the Paneth cells which are present in the intestinal epithelium that produce defensins, which also have an antimicrobial property. There are also the M cells, which actually work with the macrophages or the dendritic cells. Dendritic cells process and present antigens to the T and B cells inside draining lymph nodes. The intestinal dendritic cells also imprint naive T and B cells to express gut homing molecules, which are alpha 4 beta 7 and CCR9. These gut imprinted alpha 4 beta 7 expressing lymphocytes engage with the locally expressed MADCAM molecule, and it escapes circulation to re-enter into the lamina propria. The intestinal lamina propria contains T helper 1, T helper 17, T regulator cells. The regulator cells, T reg cells, are the cells that actually check the inflammation process and prevents unchecked inflammation. A steady state is maintained within the intestinal mucosa via coordinated activity of innate cells and adaptive immune cells. So what happens in the diseased state? For example, you have a NOD2 mutation. With a NOD2 mutation, there is a defective sensing of intracellular bacteria as well as a reduced production of defensin. The net result would be an activation of the adaptive immune response to compensate for the defective innate immunity. Now, the variant loci of ATG16L1 and IRGM genes are associated with defective autophagy. So your macrophages and dendritic cells would not work so well. Adaptive immunity can also be deranged along the IL-12, IL-23 pathway, thereby shaping the expression of the helper T cell response toward the spectrum of T helper 17 rather than T reg. The T helper response would be toward making more T helper 17 cells and therefore inciting a greater inflammation. Therefore, blocking the P40 subunit of IL-23 and IL-12, like with drugs like ustekinumab, is effective in Crohn's disease. The activation of dendritic cells and macrophages results also in an expression of TNF within the mucosa. Leukocyte trafficking is also a necessary element in amplification of the mucosal immune response. Integrins containing alpha 4 like alpha 4 beta 1 and alpha 4 beta 7, they bind respectively to the ICAM1 in the endothelium of the inflamed cells throughout the body and to mucosal specific MADCAM1, that is mucosal addressin cellular adhesion molecule 1, specific to intestinal endothelium. Now, blocking this interaction using homing inhibitors, also called integrin inhibitors like vedolizumab, natalizumab, etrolizumab, interferes with adhesion and recruitment of inflammatory cells, thereby disrupting the inflammation. T helper 9 cells perpetuate enterocyte apoptosis and inhibit mucosal healing, and you have IL-13 produced by NKT cells, further contributing to epithelial injury. Innate lymphoid cells, the ILCs, produce cytokines and perpetuate inflammation, and they are major mediators of chronic intestinal inflammation. The JAKs, or the Janus kinases, are cytoplasmic tyrosine kinases that pair with cytokine receptors, and they're essential for lymphoid cell development, survival, proliferation, and immune function. The cytokine receptor signaling, JAK-STAT pathway. Lymphoid cells rely on cytokines like IL-2, IL-4, IL-7, IL-12, IL-15, IFNs for survival and function. JAKs are non-receptor tyrosine kinases that bind to the intracellular domains of cytokine receptors. So the cytokine binds the receptor and the receptor dimerizes. The bound JAKs autophosphorylate, they get activated, and they activate the STAT proteins. The STATs dimerize, they translocate to the nucleus, and there's gene transcription. And this would regulate lymphocyte development, activation and proliferation, and memory cell formation. So by inhibiting these JAKs, we could also achieve control of inflammatory bowel disease.